how to read a scientific research paper or a scientific article. Let's first have a look at the structure of a research paper in which the authors have presented their original work. So in the top, you have the title and the name of the contributing authors. These days, this is a new trend where you would get a graphical abstract in which this picture summarizes what the study is about, a general model or idea or an illustration explaining the theme of the study. But the very, very common feature or the components of a research article are first is abstract. So when you take a paper and you want to know what is in the study, you start by reading the abstract. That gives you a brief snapshot of what this study is about. And if you are interested in the findings or if you are interested in the idea, then you read the entire paper. Otherwise, you skip that paper because you would not want to read 20 pages and not be interested in the work at all. Okay. So then comes the introduction. In introduction, the authors usually write about what is the background of the study, what information they had about the problem at hand, what are the knowledge gaps, and how are they trying to fill those gaps by the work they are doing and presenting in the current study. The next component of the paper usually is methods, materials and methods. But here you will see that methods are in the end. So write somewhere here. And you would realize that the methods are not very elaborately presented here because they are usually attached as a supplemental information. Sometimes when you are performing similar kinds of experiments, yes, you want to look at the protocols of these scientists, of these researchers, and you want to perform similar experiments in your lab. But most of the time, you're more interested in the findings. So if you want, you can go through these methods and technologies or techniques that they have used. But currently, I think they'll be beyond the scope of your class. And if you're interested, you can watch a YouTube video or read up on these techniques. For example, you can go and study how to do a cell cycle analysis, or what is flow cytometry, what is Western blotting, what are different kinds of microscopy techniques. But that is beyond the scope of the current discussion. What we need to understand is the summary or the main key points, findings of the paper. So we will be primarily focusing on the background information, what is the study about, and then I would like to walk you through these figures. In the first glance, these figures would look very complicated, but I will like to just give you a snapshot of what they are talking about in these figures. And in this 30-page long article, not every figure is important as far as your level is concerned, because some of these figures are more technical. They are supporting the other ideas or the other experiments. You just need to know an outline of what they're trying to talk about or what they're trying to explain. So I'm going to walk you through the important figures and try to establish the main conclusions of the study. After I have talked about all of that, you will have to write a summary and answer some questions I'm asking about these figures.
Now I'll walk you through how to read this paper. So if you look at this paper, the first section is summary, which is abstract, but we'll not read this the first thing, because when we'll come to the end of our discussion, we will go through this summary again and try to figure out if we have understood everything. So first we'll talk about introduction, and then we'll talk about results. So I'll explain all the figures of this paper, and you have to keep in mind that to get an overall idea of what the story is. You don't have to understand all the figures or try to get the details of all the figures. So there are some figures that we'll skip to keep our story simple. So the introduction starts with talking about GPCRs. GPCRs are G protein coupled receptors which are present on the cell membrane. These receptors help in accessing or understanding the external stimulus or the chemical changes in the environment of a cell. So scientists had found that GPCRs are absent in Trypanosoma brucei. Now we know that when trypanosomes are in the blood of its host, it's usually in the long, slender form. This form divides very actively and causes the pathology in the host. Then some material is present in the environment which is not clear where it comes from. It can be coming from the host, it can be coming from the parasite, but there is a Stumpy induction factor that induces the formation of this stumpy form. That means this long slender form differentiates into short stumpy form. The long slender becomes short stumpy. So what are the characteristic features of being stumpy? One is morphologically it's short and round. Second, they express PAD1 protein. So PAD1 is protein associated with differentiation. When long slender form differentiates into the short stumpy form, it starts expressing a lot of PAD1 protein. So you can find this PAD1 protein all over in the cell structure of trypanosome. Third important thing, that these cells are arrested in G1 phase. That means they do not undergo mitosis. They remain stuck in the G1 phase. Why? Because unlike the long slender form, the short stumpy forms do not divide, because it does not want that it keeps on dividing and then the host ultimately dies. The parasites do not want their hosts to die, because as long as the host live, the parasite has the source of shelter and nutrition. Now, although GPCRs are absent in trypanosomes, a GPR89 protein has been identified, which is involved in signaling, anion transport, and development in other organisms. This protein is present in Trypanosoma brucei, but its action is not clear. In this study, the researchers identify Trypanosoma brucei specific GPR89, and they mentioned that they were able to find that this protein is present on the cell membrane and it is involved in Stumpy induction factor signaling. This GPR89 protein also acts as a transporter for some oligopeptides and is actively involved in formation of stumpy form. So what was novel or new in this study was the researchers were able to understand the function of GPR89 and how it is involved in oligopeptide signaling, and they have mentioned that if we can target this protein, we should be able to deal with the parasite, because ultimately therapeutically we want to kill the parasites because they are causing disease in the human beings. In figure 1a, researchers have shown the structure or the tentative structure of GPR89, and they have found that GPR89 is present in the cell membrane. So if a protein is present in the cell membrane, it has the possibility of interacting with the external environment and pass on that signal to the inside of the cell, establishing a signaling cascade. This thing has been shown microscopically, where the GPR89 is shown here. You can see that it is present on the green protein is GPR89 and it is present on the plasma membrane. So it has the tendency of interacting with the extracellular environment. Because if a protein is present cytoplasmically, then it cannot interact with the outer environment of the cell. Then in figure 1c, scientists have shown that GPR89 is expressed in slender form, but not in the stumpy form. Because theoretically or hypothetically, this protein is involved in differentiation of slender into stumpy. So once it has differentiated into stumpy, its function is no longer required. So that's why the cell starts to lower down the expression of GPR89 in stumpy form, or completely eliminates the expression of GPR89 in stumpy form because it's not needed anymore. Figure 1D is not important because you need more information on understanding what are monomorphs and what are pleomorphs. Okay, in figure 1E, you need to understand one thing, that trypanosomes can be genetically engineered to increase or decrease the expression of a protein. Okay, so in this study, scientists have created a genetically engineered trypanosome from which GPR89 can be removed when they are treated with a drug called DOX. You don't need to go into the details of how these genetically engineered trypanosomes were formed. All you need to understand that in figure 1E, they are using the trypanosomes in which GPR89 can be removed when treated with DOX, or doxycycline is the name of the drug. So we see that if you treat trypanosomes with DOX, then their cell growth is halted. That means GPR89 is important for trypanosome growth. In figure 1F, the researchers found that when the trypanosomes are treated with DOX, plus DOX mean the DOX has been added to the trypanosomes. The trypanosomes are more likely to be present in G1 phase. So we have already this clue that stumpy form usually exists in G1 phase. It gets arrested in G1 phase. And here the researchers have shown that if you remove GPR89, the trypanosomes get stuck in the G1 phase. But they're not concluding anything here. They are going to further investigate what is happening to the trypanosomes when GPR89 is removed. So their findings of figure 1F is confirmed with microscopy, that once you remove GPR89 by adding doxycycline drug, the trypanosome starts to look more like a stumpy form, more rounder and shorter. So here are a few questions for you to answer based on what we have just discussed regarding the figure 1. In figure 2a, again, they have treated genetically engineered trypanosomes with doxycycline drug, and that leads to the removal of GPR89, and the trypanosomes grow slower, or their growth is significantly affected when the GPR89 protein is removed. In figure 2b, again, the researchers show that when GPR89 protein is removed, trypanosomes get stuck in G1 phase, also called 1K1N phase of their life cycle. Now in figure 1G, we saw that when GPR89 protein is removed by treating with doxycycline, the trypanosomes become short and stumpy. Again, in figure 2C, we get another evidence that after removal of GPR89, the trypanosomes become short stumpy, because remember that we had 3 clues for why we can say that one trypanosome is stumpy. One, it looks morphologically shorter and rounder. 2, they are usually arrested in G1 phase. And 3, they start expressing PAD1 protein. PAD1 is protein association with differentiation. So here they have seen that in the DOX treated trypanosomes, you see the expression of PAD1 protein. Figure 2D to 2H are not important for today's discussion because you need more background information for this. Then you have one question related to figure 2, and if you have watch the video carefully, you should be able to answer this question. In figure 3a, researchers have shown that the GPR89 protein can interact with oligopeptides. So this is an oligopeptide, and they have done this computerized simulation to see if GPR89 can actually interact with small peptides. And they have shown that there is a possibility that this protein can interact with short peptides. You can ignore figure 3b for now. And in figure 3c, researchers did a cool trick. So they took this trypanosome protein GPR89 and put them in bacteria, and they saw that when the bacteria has GPR89 protein in its membrane, it starts taking up some small oligopeptides. So they have concluded that the GPR89 might have a function as a transporter of oligopeptides. Again, figure 3D to 3E can be ignored for today's discussion. Then you have one question from figure 3. For figure 5A, the researchers took the brain heart infusion broth and treated the trypanosomes with different concentrations of this broth. They found that as they increased the concentration of the broth, the parasite growth was significantly reduced. So they concluded that there is something in this broth that inhibits the growth of trypanosomes. They also found that as the growth is getting inhibited with the treatment with the broth, there is more and more expression of this green protein, the PAD1 protein. So this green protein is the PAD1 protein. That means when the trypanosomes are treated with the broth, they, one, stop growing, and second, start expressing PAD1 protein. That means they might have differentiated into stumpy form. So what was in the broth that is making the long slender form differentiate into the short stumpy form? So the presence of PAD1 protein is confirmed by microscopy and by performing a Western blot. In figure D, researchers found that different kinds of oligopeptide sources, which can be animal peptone or tryptone or gelatin or vegetable infusion, these all peptide sources or oligopeptide sources can reduce the growth of trypanosomes. So just like what we are seeing here. So maybe in brain heart broth, there were some oligopeptides which were reducing the growth of trypanosomes. Then they found that when the growth is reduced, there is an increase in PAD1 expression. So after treatment with animal peptone or the proteose peptone or tryptone or some vegetable infusion, there is a decrease in growth along with increase in PAD1 expression. That means after treatment with certain oligopeptides, trypanosomes differentiate into short stumpy form, which stops growing and starts expressing PAD1 protein. So you have two kinds of confirmations that when trypanosomes are treated with oligopeptides, they become short stumpy. One, they start expressing PAD1 protein, and two, their growth is reduced. Because generally the trypanosomes get stuck in G1 phase when they become differentiated into stumpy form, and we know that piece of information already. So here is one question for you to guess. What was that something in the brain heart infusion that could have led to decrease in trypanosome growth? Although if you have heard the video carefully, we already have concluded what that something could be. In figure 6, researchers will use synthetic dipeptides or tripeptides to understand the chemical nature of the oligopeptides, which have significant effect on the growth of trypanosomes. So in figure A, the researchers use dipeptide and see if there is reduction. So first one is the control. There is no dipeptide in the first one. And then there are several different kinds of dipeptides, and they want to compare that compared to the first one if there are different kinds of dipeptides that can inhibit the growth of trypanosomes. In figure C, they use different kinds of tripeptides, and they want to see compared to the first one if there are some tripeptides that inhibit the growth. Now just by comparing these two figures, you know that tripeptides have more effect on the growth than the dipeptides. So that's what they have concluded in figure D, that tripeptides have stronger effect on the trypanosome growth than the dipeptides. Now these trypanosomes whose growth had been affected, they express PAD1 protein. That is interesting. So after treatment with tripeptide, which is an oligopeptide, the trypanosome growth is halted, and we know that when trypanosomes become short stumpy, their growth gets halted, and they also start expressing PAD1 protein. So that is another confirmation that we have. Another piece of evidence is the microscopy findings, in which the researchers found that after treatment with tripeptides, the trypanosomes not only get arrested in their growth, not only start producing PAD1 protein, but they also start looking shorter and rounder. And figure 6G can be skipped for now. Then you have a question based on figure 6. In figure 7, researchers have given a schematic to explain their overall idea. So they say that in the trypanosome population, there are two kinds of trypanosomes, the producers and the receivers. The producer trypanosomes produce some peptidases, and the receiver cells will receive a signal. So what is this peptidase going to do? In the environment, there are some proteins. These peptidases will chop off these proteins into small peptides or oligopeptides or tripeptides. Then the receiver cells will receive those peptides as a signal through GPR89 protein, because that GPR89 protein will be present on the surface of these trypanosomes and will help in obtaining the signal from these oligopeptides. Once that signal has been received, these cells will differentiate into stumpy form, and even the producer cells will also differentiate into stumpy form. So this is a local or a paracrine kind of mechanism in which locally some kind of cells are inducing some kind of changes or sending some kind of signal to the nearby similar looking cells. Now researchers were able to obtain a genetically engineered trypanosome cell line that produces TBPGP, which is a kind of peptidase. So they have actually made the trypanosome make more of these peptidase. Now if there is more peptidase, it will cause more chopping of the peptides, then it will send more signal to the receiver trypanosomes, and the population will start becoming more stumpy. That's the idea. But to confirm that idea, they look at the expression of PAD1 protein, and they found that when under the expression of doxycycline drug, the trypanosomes which produce a lot of peptidase, they will have a lot of PAD1 proteins, means the more peptidase is formed, the more short stumpy cells are formed. They confirmed this finding with another kind of peptidase called Trypanosoma brucei POP, P-O-P peptidase, and again they see that the cells which express more POP peptidase, they also have more PAD1 expression. So more peptidase will cause more production of oligopeptides, and those oligopeptides, when received through GPR89 protein, it leads to the induction of stumpy form. Now is the time when we can look at the summary of this paper and see if we have understood everything. So we know that trypanosome have two forms, slender form and stumpy form. The slender form divides very fast and it causes the pathology in the host. But once they have reached certain density, now they don't want to divide any further but differentiate into stumpy form that gets arrested in G1 form because it does not want to kill its host. Now, how the slender form differentiates into stumpy form has been a question, and it is shown that the slender form produces some peptidases. So slender form will release some peptidases, and these peptidases will do what? These peptidases will chop off the proteins present in their environment and generate some dipeptides and tripeptides. These tripeptides will act as the stumpy induction factor. So for a very long time, it was known that there is a stumpy induction factor, but it wasn't known whether that stumpy induction factor is a sugar, is a protein, is a fat molecule, or some different kind of chemical. It was not known. But in this study, they have shown that the stumpy induction factor could be tripeptides. Now, when these tripeptides give a signal to GPR89 protein, the trypanosomes become stumpy. And how do they confirm that trypanosomes have become stumpy? By looking at the expression of PAD1 protein and visualizing those trypanosomes under microscope. They start looking shorter and rounder,